This is lecture 9, and it's about the structure and function of DNA. So, in cells, by the end of the 19th century, they had identified nucleic acids. And we now know that DNA has the capacity to store genetic information and can be copied and passed from one generation to the next. And that these nucleotides join together with a sugar phosphate backbone. But up until the 1950s, this structure that you see in front of you was really unknown. So now we know that both DNA and RNA are what we would call polynucleotides. So they have repeating units that contain sugar, which is in blue, phosphate, which is in yellow, and then these nitrogenous bases. Now DNA has the nitrogenous bases A, G, C, or T, and RNA has the nitrogenous bases A, G, C, or U. And we'll get into that in a little bit more detail. But basically you have a polymer that's made up out of repeating subunits, and those repeating subunits code for information. So the 4 nucleotides that are found in DNA differ in their nitrogenous bases. So we have thymine, cytosine, adenine, and guanine. Uracil is found in RNA only. Now, it took quite a lot of research to figure out the double helix structure of DNA. Pivotal to that was the X-ray crystallography data that Rosalind Franklin took, and James Watson and Francis Crick were actually the first to construct a correct working model. Now, the model of DNA is a bit like a rope ladder that's been twisted into a spiral. So if you imagine on your rope ladder, the rope would be your repeating sugar phosphate backbone, and the rungs of the ladder are your A's and T's and G's and C's. And if you twisted at the bottom of the ladder, you would end up having something that looks like a helix. Now, something that you'll find in your lab that will be important is that there is something that we call anti-parallel. That means that the 2 strands actually run in opposite directions. What does this mean? Well, if you have a look at image B in the center here, what you'll notice on the left-hand side is it starts with a phosphate group, then it's sugar, phosphate, sugar, phosphate, sugar. Whereas on the right-hand side, you actually begin with a sugar and then end with a phosphate. So if you took your hands and you stuck your thumbs up, and you have your left hand with your thumb pointing upwards and your right hand with your thumb pointing downwards, that would be a fairly good example of the anti-parallel structure in DNA. For DNA to replicate, what has to happen is those 2 strands of DNA pull apart. The adenine and the thymines, the guanines and the cytosines are bonded together with hydrogen bonds, and those get pulled apart, exposing hydrogen bonds on each individual strand, and then new daughter strands are formed on top of those templates. So it's a template mechanism that allows DNA to be replicated in a fairly accurate way. Now, if the DNA replicates and mistakes are made, so where you would be expecting to get an adenine, you get some other base, that would be what we call a mutation. And that can have important consequences both for the organism that carries the mutation and for evolution as a whole. So DNA replication begins at specific sites on a double helix called origins of replication, and that replication goes in both directions and it helps to speed up the process, because if you imagined unraveling a long strand of DNA only on one side and then working your way all the way up, it would take a long time. It's like being able to unzip your zipper from 5 different locations simultaneously. It makes for a much faster replication process. Now, this picture in front of you is what we call the central dogma. It's deceivingly simple, but this is one of the most powerful images that you will see this semester. The central dogma is extremely well understood, and this pattern is found across all living organisms, and it's simply this, that deoxyribonucleic acid, also known as DNA, stores genetic information. That genetic information is then transcribed to ribonucleic acid. And that ribonucleic acid, in this picture indicated by a little red single strand, is a messenger, and it carries the message of that code outside of the nucleus into the cytoplasm. In the cytoplasm, the language of DNA and RNA is translated into the language of proteins, and the code that is residing on that strand of RNA is used to code for a sequence of amino acids. An amino acid sequence will allow you to create polypeptides, and polypeptides are the building blocks of proteins. So DNA is basically responsible for the construction of every single protein in your body. And as you learned earlier in the semester, proteins have enormous function in the body. Everything from every single enzyme for every single chemical reaction in a living organism to structural proteins, pigment proteins, etc. So any errors in DNA can result in errors in RNA and errors in proteins. Now, we understand the central dogma well enough that researchers can actually reverse engineer, and they can say something like, we have protein X that we want to make a whole bunch of. What would be the RNA sequence that would code for that protein? What would be the DNA sequence that would code for that RNA? Synthetically create that DNA sequence, insert it into a bacterium that then will grow that protein for you. Now, if you think this is absolutely wild, how many of you have eaten cheese recently? We use recombinant DNA technology to create rennet, synthetic rennet, which is used to curdle milk to make cheese. Any of your friends and family who have to take human insulin are taking a recombinant DNA human insulin that's basically made in the way in which I've described, where you've inserted a strand of DNA that codes for a certain protein into some bacteria, and the bacteria will grow it for you. So the information or language in DNA is ultimately translated into the language of proteins. It's a multiple step process. So when the DNA is transcribed, the result is an RNA molecule. When the RNA is translated, you create a polypeptide. So if you have a look at this little image, it's quite a powerful one. It says if you have AAA in your DNA strand, that would code for uracil, uracil, uracil in your RNA strand. And the uracil, uracil, uracil would code for a very particular amino acid. So we need to explore this a little further so that you can understand how to actually do this yourself. Now, the way in which RNA works is that it takes 3 bases in a row to code for a single amino acid. And there's something called the genetic code, also known as the universal genetic code, because this code is used in everything from plants to slugs to snails to humans to ants to bees. And it's simply this, that if you have a uracil followed by a uracil followed by another uracil, you will be coding for the amino acid phenylalanine. On the other hand, if you had adenine, cytosine, and uracil, you would be coding for threonine. If you had GGG, guanine, 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 you'd be coding for glycine. What you'll notice is there's not a one-to-one relationship between a codon, which is 3 bases, and the amino acid that the codon codes for. There is flexibility in this system. For example, there are at least 4 different ways to code for valine. There are at least 4 different ways to code for alanine. Now, in this genetic code, you'll notice that there are 4 codons that are very special codons. The first that you should know is called AUG. This is methionine or start. So it basically is the codon that tells you to begin coding an amino acid sequence. So all amino acids actually begin with the amino acid methionine. Now, how does the cell's machinery know when to stop making an amino acid sequence? Well, you will basically count in codons, so every 3 bases, until you hit a codon that is a stop codon. And there's actually 3 very different ways of saying stop. You have UAA, UAG, and UGA, and each of those will tell the cell's machinery now is time to stop coding. So you should always look for the AUG or the stop codons. Now, this genetic code is written in RNA language, but you should, at the end of your lab, be able to easily go between RNA language and DNA language. So, for example, the AUG, which is an RNA way of saying start, would be replaced by TAC in DNA. So a DNA sequence that says TAC in RNA would be saying AUG. And we'll get into that in a little bit more detail. So from DNA to RNA, that process requires the DNA to be pulled apart. So here you can see a blue DNA molecule, the double helix is being stretched apart. And then something called RNA polymerase, which is an enzyme, helps to create a new sequence of RNA from the template strand of DNA. So what will happen if you want to transcribe an entire gene and make that full protein? The RNA polymerase is going to look for a promoter region. After the promoter region, it's going to look for that the start sign. And once it sees the start, it's going to begin making an RNA sequence until it reaches what we call a terminator DNA sequence or a stop sequence. And that will give you your completed RNA. Now, translation, where you're going from RNA to protein, requires several different players. So translation is the conversion from the nucleic acid language to the protein language. The first thing you need is the messenger RNA. This is the sequence of RNA that tells you what amino acids you're going to need. You also need something called tRNA or transfer RNA, and this is a small piece of RNA that carries specific amino acids. So for as many different codons as there are in the universal code, there are that many different tRNA. So each codon has an anticodon, which you can see at the bottom of this tRNA molecule. And at the top of the tRNA molecule is an attachment site where it attaches to the amino acid. So basically your tRNA is like a tiny little specific Uber that brings in the amino acids to be processed and placed in the correct sequence when you are making your amino acid sequence. You also need something called a ribosome. A ribosome is the small organelle that actually makes the proteins. So what will happen is the messenger RNA will thread through the ribosome like a string through a bead. And there are these 2 binding sites that then will take the amino acids off the top of the tRNAs and hook them up together. So in this little image, you can see it a little bit better. So here's the messenger RNA sequence being threaded through the ribosome. The green cylinders are tRNAs. Their anticodons are coordinating with codons to bring in the correct amino acids in sequence, and those amino acids are being linked together at the top of the ribosome. So mutations occur when there's any change in the nucleotide sequence. So for example, and we're going to be talking a lot more about sickle cell later this semester, but if you have a sequence of DNA that's coding for hemoglobin, that's the protein that's in your red blood cells that allows you to carry oxygen. In your normal hemoglobin, one of the codons is CTT in DNA, and it codes for GAA and messenger RNA. And the amino acid that's coded for in that sequence of normal hemoglobin is GLU. Now, some people who are carrying the sickle cell allele, instead of having a T, they'll have an A in that place. And so their messenger RNA will code for GUA, and that will actually pull in the amino acid VAL instead of GLU. And that single base difference in the codon will result in a single amino acid difference in the amino acid sequence, and that is sufficient to generate a diseased state. So just think about it for a second. You have 25,000 plus genes in your body, and that is, at a minimum, the number of possible genetic disorders that human beings could have. It's crazy. Mutations within a gene can be divided into sort of two categories. They can either result in changes in the amino acids and proteins, and sometimes they don't. So you can get a base substitution. So that's a single base gets substituted, and then that changes the amino acid sequence that you have. In some cases, it doesn't affect a person's body and they're still functioning. In other cases, it does. And then there's another category which is called an indel, or an insertion and deletion. And this is actually quite disastrous, because what that does is it removes or adds a base into the sequence. And now every single codon that follows that base is interrupted. So it's changed the reading frame for the genetic message, because you're counting in threes. 3, 3, 3, 3. And now all of a sudden, in order to recover the amino acid sequence, you would have to actually remove or insert that base. Mutagens are physical or chemical agents that increase the risks of errors in DNA replication. And they include all sorts of things, such as ultraviolet light, carcinogens like compounds in tobacco and marijuana. There are many chemicals that are in our regular surroundings that are mutagens, and so we rely on researchers to provide us with good information so that we can make decisions to avoid as many of these mutagens as possible. But it's important from an evolutionary perspective to understand that although mutations are often harmful, mutations are the source of genetic diversity. And without genetic diversity, you cannot have successful natural selection, because if everything is the same, then there is no advantage to being you as an individual.